Hi everyone, my name is Talia Boston. I am currently a sophomore biology major at Oklahoma University, and welcome to my presentation. So today I will be talking about the topic of anxiety and how stress induces neuroinflammation, which then induces an anxious behavior in individuals. And there are multiple reasons why I chose this topic. So just to go in order, I wanted to destigmatize anxiety and how like some people don't take it seriously, even though there are serious anxiety disorders that are linked to neurological disorders. And I wanted to like emphasize how stress and anxiety are not linear, because I initially thought that if you're in a stressful situation, that automatically causes anxious behavior. And when in reality, that's not the case. It has to go through different pathways within our brain and different cells that have to change and malfunction, which and then creates anxious behavior. So it was kind of like a domino process that I figured out in this in my research of this topic. And so yeah, I just wanted to debunk those misconceptions, and that I also thought at first that anxious behavior was like a voluntary response, when in actuality it's an involuntary response, and that there are many neurological intermediates within anxious behavior. So I wanted to clear up my knowledge on what I previously knew about anxiety and dig deeper into how what happens neurologically and how it links to it behaviorally, and what we can do as scientists to alleviate the major symptoms of anxious behavior. So to get into the table of contents, I will talk about four major points. My first point will just be giving a brief background / introduction. And within these slides, I will talk about the three common types of anxiety and the type of organs and cellular processes in the central nervous system that cause anxious behavior, or in part of the part that are in part of the pathway of anxious behavior. Next, I will go into the type of cells that are in this pathway, such as microglial cells, talk about what they are and how they're important in inducing anxious behavior. And then I will link this topic to an experimental study done on model organism of rats on how repeated stress causes anxious behavior. And lastly, I'll just wrap it all up and talk about its importance, what you can take from my presentation, apply it to your real world situations, and how we can delineate and destigmatize the topic of anxiety. So first, like I said, I'm going to talk about the brain more specifically and how anxious behavior is related to different functions of the brain. And before I go into that, I wanted to talk about the three common types of anxiety. So the first type of anxiety is called general anxiety disorder. And as you can tell by the name, it's not as serious as one might think it is. So the type of trigger stress that causes this type of anxiety disorder is acute stress situations. So what I mean by acute stress situations, I wanted to highlight that in these type of situations, if you were put it on a spectrum, it's not as extreme as other anxiety disorders that we'll talk about further. And it is caused by recent major events. So if I were to give an example, say like a school example, I feel like we all can relate to this. Say you have an exam the next day and you're very stressed about it. You've been stressing about it for weeks. You have been preparing. You don't know if you're going to do well on it. And so with this type of stress, like predicting the future, it causes concentration issues and disruption in your sleep cycles. And because it causes concentration issues, you're hyper fixated on this one thing that you're worried about. So it's excessive worry and anxiety that you won't get over this type of anxiety until that event passes. So it doesn't have any long-term effects. It's more situational. That's why it's called acute stress. Now, in panic attacks, it's more broad, as that's the type of panic attacks people experience depend on what they've been through throughout their life. So that's why it's triggered by episodic stress. And it is caused by a series of stressful events. So say you've been having like a rough time with like your personality, like you don't know who you are. So you're just stressful with that. And then you're put in a situation where people challenge your personality and your body just normally doesn't know how to react to this type of stress. It causes physical panic where you're doing like heavy breathing, your heart rate increases, and you can't really focus on where you are at the moment. And so people try to downplay this type of anxiety disorder because in when this person is experiencing this type of anxiety disorder, they're experiencing an excessive amount of terror when there's no actual real physical danger present that's attacking them. They're just freaking out and they don't know how to respond to the situation that they're in. So it's kind of hard to define and is very different among our population depending on the person and the type of stress that they've been under. And then lastly, the third most common type of anxiety is PTSD or post-traumatic stress disorder. And I feel like everyone is very knowledgeable about this type of anxiety disorder as it is related to war veterans and what they experience when they come off of the battlefield. And the type of stress that causes this type of anxiety disorder is chronic stress because of the seriousness of the trauma that they went through. So personally, my dad, he was a veteran back in the 90s and he still does experience PTSD every now and then. And it is triggered by certain noises, sounds, words. And it can be the littlest things that sends him back to that moment where he was, in the battlefield under extensive pressure and stress. And it's hard to distinguish reality from what's not real or from the past and the present. So this is the most extreme type of anxiety disorder. And those type of anxiety disorders and type of stress that triggers it relates to how it is processed in the brain. So the type of system that is in control of the type of emotional processes it relates to the behavior aspects is the limbic system. And the limbic system is part of the central nervous system. And within the central nervous system, you have different organs such as the different lobes. And so right here, I wanted to highlight the frontal lobe. And with the frontal lobe, it is in charge of emotional processes, which again is linked to the limbic system and decision-making skills. So as you can like predict, that does relate to anxious behavior, because when you're under stressful conditions, you can't really make logical decisions or abstract decisions. And I also wanted to highlight the amygdala because it also isn't in charge of our type of response under stress. And more specifically, it elicits a fear response, which I'll talk about in the next slide. But basically in this slide, I just wanted to highlight that it responds to external stimuli from our environment, and the type of stimuli that we're talking about here is stress, of course. And so it consists of certain receptors that notices the type of stress in our environment, and it has to decide how it react to it. And so this is where the amygdala comes in and how it reacts to the type of stress that we undergo. The number one emotional response we elicit is the fear response. And here I just have the image of the amygdala, and the pathway is sends the external stress stimuli to, and what that organ elicits to our body physically and behaviorally. So say the amygdala, the receptors in the amygdala experience a stressful stimuli, it sends it, for example, to the lateral hypothalamus, and what it does, it increases heart rate and high blood pressure. And I said that it kind of elicits the type of fight or flight instinct because when you're under a stressful situation, you have either those two choices to actually act upon it or become in freeze mode. And when you become in freeze mode, the type of pathway it goes through is the central gray area of the brain of the central nervous system, where you're not able to socially interact with others. So basically what the amygdala wants to do is to create a fear response in the body and allow it to properly react to the type of stress it is exposed to. And what is not highlighted in the last slide is that in that pathway, it also manipulates the type of cells in our central nervous system. And the major cell that causes anxious behavior is the microglial cells. And right now I'll talk about the importance of it and the structure of it and how it changes. So what are microglia? So I included two different images to give you a visualization of what microglia look like. And so basically what microglial cells are, they're an abundance of cells that are distributed throughout our spinal cord and our central nervous system. And they are innate immune cells that are responsible for reacting to different factors, different external factors. And more specifically is responding to stressful factors. And the way it looks like is that I think of it as our hand as the central nucleus. So like this is the central nucleus and our appendages are the branches that stick out. So scientists classify this as highly ramified, which basically means that it has a very complex structure. And the reason I included this image, which shows microglial cells, astrocytes, and neurons, is that although they look very similar, they have different functional features. So if you look at the green microglial cell here, it has a smaller central body in comparison to the astrocytes, and the branches of the astrocytes are more jagged, and with the microglial cells is more smooth. And this is directly linked to the function that they do. And the next, and the neurons, they're more elongated because they consist of axons and dendrites. And now that I brought that up, the microglial cells do interact with neurons, and its job is to create a homeostatic environment of reuptaking certain neurotransmitters such as dopamine and serotonin. And when it creates a homeostatic environment, it creates a perfectly balanced state in our central nervous system, so we don't experience mood changes or mood swings. But if this is not maintained and the microglia doesn't function properly in relation to the neurons and doesn't have a stable relationship with the neurons, it does undergo morphological changes. And with the morphological changes comes with an indirect effect on the immune system. And like I said here, it releases cytokines, which is in relation to the immune system. So when the morphology means the shape, when the shape of the microglial cells are changed, which I'll talk about in the next slide, it releases cytokines, which is part of the immune system. So like I hinted at in the last slide, when we do undergo stress, our body is basically in a state of shock, and it is trying to process what is going on in our environment and relate it to what's going on internally in our cells. So what is manipulated here is the branching of the microglial cells. So here I attach an image from a random experiment I saw online, and I liked this experiment because it showed visually what happens to the microglial cells under different types of stress. So that's why I introduced what the different types of stress are, so you know what we're going to be talking about here. So here, if you look towards the left arrow, it shows what happens to the microglial cells when it just induces a quick shock, a short-term type of shock, which is acute type of stress. And as you can see here, in comparison to the image up top, the branches of the microglial cells shrink drastically. And when it shrinks drastically, it increases from pro-inflammatory cytokines. And if you look to the right arrow, this is a different type of stress, such as chronic stress, like we talked about before. And it's different than acute stress, as the branches don't shrink and actually elongates and becomes even more ramified than it was before, which is a problem because it also releases an abundance of pro-inflammatory cytokines. And so if we were to sum up what I just said, long branches equals chronic stress and stunted branches equals the cause of acute stress. And the reason why it's such an issue is because it releases so much pro-inflammatory cytokines in comparison to anti-inflammatory cytokines, because when it creates more pro-inflammatory versus anti-inflammatory, the central nervous system is in a state of homeostatic imbalance, and it's not able to process the stress functionally. And like I said, the amount of neurotransmitters are not in correct balance like as it should be. And this directly relates to an experimental study that wants to validate this type of observation in our microglial cells. So the study that I will be talking about is how repeated stress induces a pro-inflammatory state, increases amygdala, neuronal, and microglial activation, which then causes anxiety in adult male rats. So where this experiment was conducted, it was conducted at Rosalind Franklin University in November 2019, and it was established by various authors, but most importantly, Munshi. And what he wants to figure out here is whether a specific type of stress called repeated social defeat stress. Sorry, it's kind of like a mouthful. Repeated social defeat stress, or RSDS for short, produces a pro-inflammatory state in the CNS due to altering the function of microglial cells critical to influencing behavior. So they want to analyze the different functionality features of our cells and our organs that play a part in relaying anxious behavior. And here the model organism will be rats. So to go over the methods and how this experiment was designed, they wanted to experiment on two different type of rats. And those two different type of rats are Sprague-Dawley rats and Locke-Evans rats. So to give these rats some characteristics, we have here in the right-hand corner, the Sprague-Dawley rat, which is the white-coated rat. And below that picture is the Locke-Evans rat, which has a black and white coating. The reason that they are different is because Sprague-Dawley rats are known as the more aggressive rat in comparison to the Locke-Evans rats. And this is a key feature because they are testing repeated social defeat stress. So they want to see if they put these two rats in close proximity, how they react with each other. So what they do is they gather up 149 Sprague-Dawley rats, male specifically because females will not produce the amount of T cells that they want to analyze and cytokines. So they will only want to use male rats. So I just want to highlight that. So what they want to do is that they put the Sprague-Dawley rat and the Locke-Evans rat in one enclosed cage. And they don't put any dividers between them because the purpose of it, they want them to interact with each other. So they let them interact for about 15 minutes. They leave them alone. They don't give them any type of reward or food. They're just interacting with each other. And the Locke-Evans rat is characterized as the intruder. So they first put the Sprague-Dawley rat in and then the Locke-Evans rat to see if the Sprague-Dawley rat will get irritated and then retaliate against the Locke-Evans rat. And another thing I wanted to say is that they wanted to keep the experimental rooms dimly lit because rats are extremely sensitive to light. And if they're extremely sensitive to light, they will go to sleep, which is kind of counterintuitive, where usually in light, someone wants to stay awake, but in relation to rats, they want to go to sleep. So they want to keep the experimental rooms dimly lit so they can function properly and get accurate results. And so like I said, they let the rats interact with each other. They leave them alone for about 15 minutes and they characterize a social defeat when the Locke-Evans rat lays on his back with his stomach facing upwards. So it's just laying on his back. And what this says to the scientists is that I've given up. I can't retaliate anymore. I do not want to fight with this other rat anymore. I am defeated by this social stress. And how they want to analyze these findings is through a series of behavioral tests. So after 48 hours have elapsed after the experiment, they want to start collecting their data. So the different type of behavioral tests that they want to conduct is the open field test, the social interaction test, and the elevated plus maze test. So in the open field test, you can kind of tell what they want to do based off on the name. So they put the Locke-Evans rats, the test subject, in a very large box, an open area, no top on the box. They place them in there, or they place IR sensitive cameras because they also want to keep this experimental room dimly lit, just as they did in the other experiment. And what they want to do is to see the parameters where they want to go in the box. Do they go towards the corners? Do they stay in the middle? They just go around in a circle? How are they acting behaviorally, which will then tell us that they do show anxious behavior. And then the social interaction test, they want to see how this test subject reacts with the novel subject. So in this situation, the test subject is the Locke-Evans Long Evans rat, sorry, that's also a mouthful, and how it reacts with the Sprague Dolly rats. Do they nudge against each other? Do they sniff against each other? Do they box against each other? And what boxing means is that they like nudge like their heads against each other, which is kind of interesting because I couldn't really imagine rats boxing, but that's how they identify it as. So they want to see how those two subjects react with each other. And if they react socially a lot, then they don't show anxious behavior. But if they do less, they don't react much with each other, then they show anxious behavior. And then lastly, with the elevated plus maze test, they use a T-shaped maze. It's called a T maze. And it consists of 2 open arms and 2 closed arms. So in the 2 open arms and the 2 closed arms is characterized as if they spend more time in the closed arms that they experience anxious behavior. And if they spend less time in the open arms that they experience anxious behavior or relay experiences anxious behavior. And they've just put the Long Evans rat in there and they let it wander. They don't also, like I said, they don't give it any type of reward. This isn't a type of learning experiment. They just want to see what the rat naturally does. And after all the behavioral tests are conducted, they also want to relay what happens morphologically with the microglial cells. So they use a type of imaging technique that they don't go in much detail about, but they want to show how it changes after the Long Evans rats experience social defeat stress. And they also want to collect their blood cells to mark the type of cytokines that are increased, the one that stays stagnant, and how those compare to each other. So these are the 3 behavioral tests that I talked about that I wanted you guys to visualize, just in case you were having a hard time trying to figure out what they look like. So to the very far left, that is the open field test. Like I said, they put the Long Evans rats in a large open box, let them do their thing. Basically, they let them wander around, get used to the surroundings, whether they stay in the center. So they're basically measuring the distance, how much distance they cover within this experimental room. Next, we have the social interaction test where they're trying to see, like I said, you see here that they're nudging against each other, sniffing against each other. And so in this case, they would not show anxious behavior because they're comfortable with being around each other. Whereas if they weren't, if they were there's a lot of distance between them, then they would show anxious behavior. And then the T-shaped model, like here, these are the 2 open ends. And then on the sides, those are the 2 closed ends. And with this rat specifically, it's more in the open end, so it wouldn't show anxious behavior. It likes staying on the open. And you would compare this to humans. Like if a human wants to stay in their house all the time, maybe they have social anxiety. They don't want to be around people. Whereas if they were out in the open in social settings at parties, they're not anxious about their environment. They're very comfortable about where they are. So, and there you can see the relation. So now I'm going to talk about the results and what they concluded from these results. So there's a series of graphs here, and I wanted just to highlight the graphs that have significant results. So I put a red box around graph C. And like I said, this experiment was conducted over the longevity of 5 days. And here they calculated how many rats submitted and how many didn't submit. And there's a huge difference in how many rats submitted versus how many didn't. And so, for example, in day 1, about like 58 Long Evans rats submitted. But I wanted to point this graph out specifically because I wanted to show that there were rats that didn't submit, because you would think that all of them would just give up, which shows that some of the Long Evans rats are more stubborn than the others, and they are up for a fight, so they are not susceptible to anxious behavior. So because those rats didn't submit, they didn't use them for the behavioral test, which is also I wanted to make known. They only used the rats that submitted due to the social defeat stress to take results from and to take data from. And these 2 graphs don't really show anything about anxious behavior. They just show that there's an average number of attacks over the 5 days, and the amount of submissive behavior is average over the 5 days. So, but it's a graph C that showed the significant amount of data in these 3 graphs. And next, these are the data from the different behavioral tests. So in the open field test, I put a box around the two first graphs. And before I go into different features and what they concluded, I wanted to point out the different aspects of the graph. So this star right here, that just means that it had a significant difference and a significant result from this evaluation. So these graphs, both of these graphs do. So as you can see, this one is measuring the amount of distance they cover in the open field test. And in the control group, they wander around very averagely in the open field test, whereas in the repeated social distance group, which is the experimental group, they have a high standard deviation, which is shown by the whiskers here, which is that some of them do go around the box with a high distance where some of them don't. And if they don't, like you see, one is all at the bottom, like close to zero, that means that they show anxious behavior. And in the second graph, you can see even more difference, whereas the control group covers more distance, whereas the experimental group barely covers any distance, which then shows anxious behavior. In the social interaction test, there's a lot of significant differences between the control group versus the experimental group, as there is a high number of interactions in the social interaction test and the control group because they didn't submit, but in the group where they did submit, there's a low standard deviation, and that just shows that they wanted to stay more to themselves, which then shows anxious behavior. And then lastly, in the elevated plus maze test, as we predicted, the more time they spent in the open arms, then the less anxious that they would show in their behavior. So in the experimental group, they spent less time in the open arms, and the scientists predicted that if they did that, then they showed anxious behavior. And then another graph that I thought had a lot of significant results was the collection of the cytokines. And cytokines, another word for cytokines are interleukins, and they are part of the immune system, which produces either a pro-inflammatory state or an anti-inflammatory state. So if you were to compare these two graphs, there is a significant increase in the interleukin 1B and interleukin 1C. And like these also, like I said, these also have stars above them, which show there is a significant difference in the amount of the number of cytokines it releases due to the change of the morphology of the microglial cells. And as you can see in the anti-inflammatory graph, it was stagnant among these three interleukins, which shows that when one undergoes a lot of stress, the anti-inflammatory response is basically muted. And what they want to do is to create an inflammatory response to signal to the brain, hey, like we're in danger. I don't like the situation we're in. We need to react to it as soon as we can. And like I said, this does interrupt the morphology or the shape of the microglial cells. So here I have three images. I really only wanted to talk about two, which is the ramified image of the microglial cells versus the amyloid image. So if you think about the ramified image as an uninterrupted, functional, efficient microglial cell. And here it has a very stable nucleus with a lot of branching, which means that it's ramified. But when it undergoes social diffuse stress, which is a type of chronic stress, it basically shrinks. So that's why it's called amoeboid, because it's its own like entity. The branching is very little. And because the branching is very little, it cannot function in relation to the neurons and eliciting neurotransmitter in a hormonal imbalance, in a hormonal balance. So now that I've talked about those major findings of the experimental study, I wanted to wrap it all up and talk about the major key points you can take from it and how we can use this in future instances in pharmaceutical developments. So what I wanted you guys to take home from this, or just to wrap your heads around, is that stress is not a direct process. It is basically opposite of that. It's an indirect process through where when you are in a stressful environment, it has to go through certain neurological factors to then create an anxious behavior in humans or in other animals like rats that we just looked at. So firstly, the stress response leads to neurological changes. And like we said, the morphology of the microglial cells change as the branching either increases or decreases or elongates or stunts. And because this happens, it creates cell malfunction. So the type of stress that we looked at was chronic stress. So this type of stress created cell malfunction in microglial cells and how it reacts with neurons and between the axons and dendrites. And when the microglial cells are affected, it then sends a signal to the immune system that, hey, homeostasis is messed up. We need to fix it. So then it creates a pro-inflammatory state in the central nervous system by increasing the amount of cytokines in the immune system. And because it creates a pro-inflammatory state in the central nervous system, there's an increased balance of hormones, which then shows mood swings in individuals, which then leads to even more serious neurological disorders. So as you can see here, it's kind of a domino effect. One leads to the other, which leads to the other, which leads to an even serious behavioral aspect of someone. So they have to deal with it. And because it is so dynamic and is all connected with each other, it is extremely important, and we have to be able to control it through pharmaceutical means. And through this, I emphasized many times that the homeostasis regulation is threatened. And because the homeostasis regulation is threatened, like I said, it causes a lot of depressive disorders, not just anxious disorders, because the amount of anxiety can only reach a point where it gets even worse and causes even worsening neurological disorders. So this is a huge issue, and it can inhibit a person from reacting functionally in society and just basically stunts their social interaction growth. So I wanted to talk about a certain receptor that is part of the amygdala, not to go too much detail because I am wrapping it up. I just wanted to mention how there's this one receptor in the amygdala that if we block this type of receptor that responds to external stimuli such as stress, that we can alleviate the dramatic effects of stress on hormonal imbalance and mood swings. And with this, we can make very large pharmaceutical advances, as we can create drugs that target specific stress, like we talk about episodic stress, acute stress, and chronic stress, to keep those three in mind. And if we can target those specific type of stress, we can alleviate the dramatic effects of stress on anxious behavior. So that is my presentation, you guys. I hope you guys learned a lot. And I hope you guys can take this information, apply it to your everyday lives, and educate others on the dramatic effects of anxiety and how it affects our neurological functions in the central nervous system. Thank you.